The Pictish Problem, produced by D.W. Drafted. In his work called The Ecclesiastical History of the English People, the Venerable Bede wrote about how the Picts began, as people understood it, in 731 CE. Right now, there are 5 languages spoken in Britain. These include English, British, Irish, Pictish, and also Latin. In the beginning, everyone living on the island were Britons. The island got its name from them. They traveled to Britain by sea, people say, from the region of Armorica. They claimed the southern section for themselves. Once they had taken control of most of the island, starting from the south, a story tells us the Pictish people left Scythia by ship. They sailed into the ocean with just a few warships. The wind pushed them past the farthest edges of Britain. They reached Ireland and came ashore on its northern coast. The Picts requested a place where they could live. The Irish replied that the island couldn't hold both groups. But they said, "We can offer you helpful guidance about what you should do. We're aware of another island close to ours, toward the east. We see it often in the distance when the days are clear. If you travel there, you can build a home for yourselves. But if anyone opposes you, call on us for support." So the Picts traveled to Britain. They began to settle in the northern sections of the island, since the Britons had already taken the southern areas. Because the Picts had no wives, they requested some from the Irish. The Irish agreed to provide women, but only under one condition. In any disputed situations, they must choose their kings through the female royal line instead of the male. And everyone knows that the Picts have followed this tradition until today. 2,000 years ago, the northern British Isles belonged to the Picts. The Romans arrived and departed. They invaded the British Isles, but never conquered Scotland. Hadrian's famous wall stands as proof of that. Then the Church arrived. After them came the Angles, Saxons, and Vikings. By the Norman conquest, the ancient Picts had already become a legend. They were a nearly mythical group, more connected to Celtic fairies and druids than to an actual pastoralist federation of different tribes and ethnic groups. But what became of the Picts? Where did they disappear to? Over thousands of years, the descendants of the original Picts have vanished into history. For many generations of scholars, this has been called the Pictish problem. Only recently, with modern technologies like DNA testing and oxygen and strontium isotope studies of the remains we still have, plus the current inhabitants of lands the Picts once controlled, have we started to solve this ancient puzzle. Released in 2023, this open-source article in PLOS Genetics offers some first answers to the Pictish problem. The text has been modified and adjusted for general readers. There's a link to the original research in the description. Imputed genomes and haplotype-based analyses of Picts of early medieval Scotland reveal fine-scale relatedness between Iron Age, early medieval, and the modern people of the UK. Abstract. There are old questions about where the Picts of early medieval Scotland came from and who their ancestors were around 300 to 900 CE. These questions arose partly because of strange medieval origin stories, their mysterious symbols and writing, and the limited written records. The Picts, first written about in the late 3rd century CE, fought against the Romans. They later built a strong kingdom that controlled a vast area in northern Britain. In the 9th and 10th centuries, Gaelic language, culture, and identity took over. This changed the Pictish realm into Alba, which came before the medieval kingdom of Scotland. So far, no complete study of Pictish genomes has been released. Questions about their biological connections to other cultural groups in Britain remain unanswered. Introduction. The genetic origins of today's UK populations have been widely researched. They can mostly be explained as a mix of 3 ancient genetic backgrounds that mirror Western European ancestry. These include Western European Mesolithic hunter-gatherer ancestry, early European farmer ancestry that came from Anatolian Neolithic farmers, and late Neolithic steppe-related ancestry. Our knowledge of more recent population changes in the British and Irish Isles, meaning Britain, Ireland, and nearby smaller islands, has grown through large-scale ancient genome studies. These studies reveal major gene flow from mainland Europe into southern Britain during the Middle Bronze Age. This created genetic differences between Iron Age groups from southern and northern Britain. Today's genetic variety in Wales, Cornwall, Devon, and Western Ireland shows long-lasting genetic patterns. These may have already existed during the Iron Age, but the shortage of ancient samples, especially from Scotland, stops us from directly testing this idea. Pockets of older ancestries might have survived in isolated regional populations for long periods. The British and Irish Isles experienced complicated cultural changes from the Iron Age to the early medieval period. The Romans controlled part of Britain down to southern Scotland from 43 to about 410 CE. However, the only autosomal DNA research on Roman Britain suggests this occupation caused very little detectable gene flow from mainland Europe. Many episodes of long-distance migration across Western and Central Eurasia increased during the late ancient period, roughly 300 to 800 CE. This happened before and after the Western Roman Empire collapsed. In Britain, Angles, Saxons, and other Germanic-speaking groups probably came from Scandinavia, the Low Countries, and parts of Germany. They settled mostly in southeastern and central Britain. Genetic evidence shows widespread mixing with local populations who carried genetic ancestry from the Iron Age. During what's called the Viking Age, starting around 800 CE, Scandinavians settled in the Danelaw in northern and eastern England. They also settled in coastal areas of Ireland and northern and western Britain. This led to mixing with the people of Ireland and western and northern Britain over nearly 4 centuries. Additionally, local culturally separate groups lived in Britain around the end of the Roman period, before major Anglo-Saxon settlement. The Britons, who spoke the ancestor language of Welsh and also Latin, lived on the island south of the Firth of Forth. The Gaels of Dalriata occupied Argyll and the southern Hebrides in Scotland. The Picts lived in the rest of Britain north of the Forth. The genetic variety between and within these groups is poorly understood. Particularly, the lack of genomes from Scotland has limited our ability to grasp how the genetic structure changed between the Iron Age and the early medieval period. Among the peoples living during the first millennium CE in Britain, the Picts, around 300 to 900 CE, are among the most mysterious. Their unique cultural traits like Pictish symbols and the shortage of direct writing have led to many different theories about their origin, lifestyle, and culture. This is the so-called Pictish problem. Other than a list of kings and hard-to-understand Ogham alphabetic inscriptions, the only written records come from their neighbors. The Romans, and later the Gaels, Britons, and Anglo-Saxons wrote about them. This lack has been made worse by a thin archaeological record with few settlements and even fewer burial sites from this time. In modern times, ideas about Pictish origins have changed, often based on cultural and political biases. The Picts and their languages have been considered Germanic, Gaelic, Brittonic, Basque, and Illyrian, among other theories. In the 1950s, Jackson influentially claimed that the Picts spoke a non-Indo-European language and a Celtic language similar to ancient Gaulish. The current agreement is that they spoke a Celtic language closest to that of neighboring Britons. Cornish, Welsh, and Breton come from this language. However, some still argue from undeciphered inscriptions and other words that some Picts spoke an otherwise unknown language. This would presumably come from a pre-Celtic population. This theory is supported by recent findings that argue late Bronze Age migrations in southern Britain may have brought Celtic or Celtic-related languages to this area. However, these migrations seemingly didn't influence northern Britain as much. So the question stays whether the Picts were somehow fundamentally different from their neighbors. In the medieval period, the Picts were thought to be immigrants from Thrace, north of the Aegean Sea, Scythia in Eastern Europe, or islands north of Britain. However, Irish accounts and the Northumbrian scholar Bede added that before settling in Britain, the Picts first got wives in Ireland under one condition. Pictish. Succession would pass through the female line rather than the male line. This is where the theory that the Picts practiced a form of matrilineal comes from. Succession and perhaps inheritance would go to the sister's son rather than directly through the male line. However, our earliest source for this practice, Bede's Ecclesiastical History of the English People, finished in 731 CE, stated that it was limited to occasions when the succession was disputed. It's now argued that the origin legend was meant to strengthen Pictish identity, and make certain kings whose claim to the throne came through their mothers seem legitimate. Still, matrilineal remains one possible explanation for the absence of father-to-son succession in at least one Pictish royal dynasty before the mid-8th century. Here, we aim to give new insights into the genetic variety of the Picts through analysis of two ancient whole genomes. These were sequenced from individuals dug up from two Pictish-era burial sites, Lundin Links in southern Pictland and Ballintor in eastern Ross, northern Pictland. We imputed diploid genotypes alongside published medium to high-coverage ancient genomes. These include individuals from England dating from the Iron Age, Roman, and early medieval periods. We co-analyzed these with previously imputed ancient genomes from the Orkney Islands. These date from the late Iron Age, Pictish period, and Viking Age. Using allele frequency and haplotype-based methods, we aim to figure out the genetic relationships between the Picts and neighboring modern-day and ancient populations. Additionally, using the mitogenomes of 7 individuals from Lundin Links, we explore how differences in female mobility might be due to possible post-marital residence customs like matrilocality or female endogamy. These may have shaped the genetic variety at this possible high-status cemetery. We discuss what this means for our understanding of Pictish elite descent systems, DNA extraction, sequencing, and radiocarbon dating. We retrieved DNA from 8 individuals. 1 came from Ballintor in Easter Ross, and 7 from Lundin Links in Fife. These represent the northern and southern parts of Pictland. 10 samples from Lundin Links and 3 from Ballintor were radiocarbon dated to the 5th to 7th century CE. The individuals lived during the time that Pictish identity existed. They're from areas likely to have been within Pictish territories around 700 CE. Analysis of uniparental genetic markers. The mitochondrial haplogroups found in the samples are common in today's northwestern Europeans. The subclade J1C3 was identified in 3 individuals out of 8. For paternal Y chromosomal lineages, we assigned the 4th individual from Lundin Links, Lund004, to R1b DF49. This is mainly found in the UK and Ireland. It's a subclade of R1b P312S116 haplogroup which Belgica peoples brought to Britain during the Chalcolithic period, along with steppe-related ancestry. During the Chalcolithic, R1b-derived haplogroups mostly replaced the dominant I2a Y chromosome lineage from the British Neolithic. The exception was Orkney, where I2a continued into the Bronze Age. R1b subclades are extremely common across Britain and Western Europe from the Iron Age onward. Allele frequency-based genomic affinities in ancient Britain. To investigate population connections of the individuals from Pictland, we did principal component analysis and admixture analyses on a dataset including today's Europeans. The PCA shows that the ancient individuals from Britain generally fit with today's individuals whose 4 grandparents lived in Britain. However, we noticed some variation among these individuals. BAL003 and Lewin004 fall within the modern Welsh cluster, but BAL03 is noticeably closer to the present-day Scottish, Orcadian, English, and Northern Irish clusters. This suggests some genetic differences among individuals from Pictland. The Iron Age and Roman period individuals from England are spread across the modern English, Northern Irish, Scottish, and Welsh clusters. 4 ancient Orcadians from the Iron and Viking Ages fit with today's Welsh, Northern Irish, and Scottish populations. However, 2 Viking Age Orcadians are intermediates between the British and Scandinavian clusters. This agrees with previous results, finding evidence of mixing in these individuals between British-like and Scandinavian-like ancestries. The early medieval individuals from England are intermediate between modern English people and Scandinavians. This matches various degrees of mixing between Iron Age groups from England and immigrants from northern Central Europe. These results agree with the pseudo-haploid-based analyses of the genomes. They show broad connection to modern Western Europeans, but with much better detail. Haplotype inferred genetic structure in early medieval Britain. Haplotype-based methods have been shown to perform better than conventional unlinked SNP approaches in finding population substructure. Our analysis shows that the genomes from Lundin, Lynx, and Balintore form a genetic cluster together with genomes from the Iron Age and Roman period from England. The exception is 6-Drift3, who instead shows strong connection to Western Central Europe or Scandinavia. Also included are genomes from the late Iron Age to Viking Age from Orkney. The exceptions are VK204 and VK205, who carried substantial Scandinavian-like ancestry. This cluster also includes Viking Age individuals from Britain, Iceland, and Scandinavia. The latter likely represents individuals buried in Scandinavia, but whose parents were from a British-like gene pool. Based on outgroup F3, the individuals from Orkney, Scotland, and England, dated from the Iron Age to medieval period, are symmetrically related to each other. However, we also note that BL-DWF3, but not LUN-WF4, shows multiple cases of genetic sharing with early medieval individuals from England. This implies substantial shared ancestry and possibly recent gene flow from a source genetically similar to those samples. We therefore suggest that individuals from Pictland shouldn't be thought of as a uniform genetic group. Instead, they're a complex mixture of contemporary genetic ancestries. The unlinked approach used in the admixture analysis also reveals a minor but detectable genetic structure. While the proportion of ancestry components are similar across BAL003, LUN004, Iron Age, and the Roman period in England, the late Iron Age and unadmixed Viking Age Orcadians are different from this group. They show an absence of the grey and green ancestry components. These were likely first brought by Scandinavian migrants, as they're first seen in VK204 and VK205, and then in modern Orcadians. These components are also carried in high proportion in modern Norwegians and Danes. Iron and Viking Age individuals from Orkney without Scandinavian-like admixture share more genetic drift with modern Orcadians. The results confirm the relatively high proportion of shared genetic drift in Orkney for the last 2,000 years. Nevertheless, a high relative count of identity by descent sharing between LUN004 and late Iron Age or Viking Age Orcadians shows that gene flow between Orkney and mainland Scotland likely happened, or a source outside Scotland that also contributed ancestry to LUN004 likely occurred. The Iron Age and unadmixed Viking Age Orcadians also show the highest degree of a red ancestry component. This is inconsistent with having come from direct gene flow from any population included in this study. Instead, it likely reflects keeping a less diverse pre-Iron Age ancestry in Orkney, or strong genetic drift like a bottleneck or founder effect. In fact, recent research showed that Bronze Age populations in Orkney were different from their counterparts on mainland Britain because of strong genetic drift. This resulted from a small ancestral population size, which was figured out from seeing a relatively high number of short runs of homozygosity. The Bronze Age Orkney population also kept the Y chromosomal haplogroup I2, which is linked with Neolithic ancestry. Meanwhile, the R1b haplogroup, linked with Bell-Beaker expansion, largely replaced the I2 haplogroups in the rest of Britain. Moreover, although modern Orcadians are different from the rest of the British Isles due to extensive mixing with Scandinavians, recent genomic research shows that genetic drift also played an important role. This matches our results that show a high proportion of shared identity by descent segments among modern Orcadians. This means they share a high proportion of recent common genetic ancestors relative to most modern European populations. This is typical of small or genetically isolated populations. Overall, these data indicate a long-term small population size, which likely contributed to the extensive genetic drift observed in modern Orcadians. The genetic differences between populations living in Orkney and Scotland during the late Iron Age and early medieval period could thus be partly explained by different degrees of genetic drift. Analysis of genetic continuity across Britain. The Pictish data allow us to get a cross-section of Iron Age and early medieval genomes across Britain. We can directly look at the pattern of haplotype sharing between them and today's genomes. The Iron Age and Roman period individuals from England and Scotland, except 6-3, share more identity by descent segments with today's individuals from Scotland, including Orkney, Northern Ireland, and Wales. They share more with these groups than with any other European populations included in our analyses. This matches the structure seen in the PCA analysis. We also show that all early medieval individuals, excluding I-177, share more identity by descent with modern Danish than with any other present-day population. This suggests genetic continuity between modern-day Danish and the ancestors of these individuals. The analysis also revealed high identity by descent, sharing between early medieval individuals from England and today's people across Britain. This follows a southeast to northwest gradient. This pattern suggests that northern continental European ancestry, linked with Anglo-Saxon migrations, expanded out of southeastern England. This was followed by mixing with local populations. This scenario matches previous research. BL003 and Lund 04 share a high proportion of identity by descent segments with today's people from Western Scotland, Wales, and Northern Ireland. This is similar to the individuals from late Iron Age Orkney and England. However, unlike these individuals, Lund 004 and to a lesser extent BAL003 share fewer identity by descent segments with the present-day Eastern Scottish population sample. Previous research suggested that the genetic structure between Western and Eastern Scotland could result from the divide between the kingdoms, the Gaelic-speaking Dalriata in the west, and Picts in the east. This seemingly contradicts the results presented here. Instead, the present-day genetic structure in Scotland likely results from more complex demographic processes. These can't be reduced to a single model. We propose two non-exclusive processes that might explain the observed pattern of identity by descent sharing between the Iron Age and early medieval populations and today's Scottish population. The first is substantial mixing from immigrants who brought Iron Age Orcadian and England-like ancestries, likely independently. This partly replaced the Eastern Scottish early medieval gene pool. Indeed, in the following centuries, 1100 to 1300 CE, Eastern Scotland received substantial immigration. This included settlers from Britain, south of the Forth, France, and the Low Countries. Under this scenario, BAL003 and LUN004 are good representatives of the broader ancestry present in Scotland during the Pictish period. Alternatively, the ancestors of BAL003 and LUN004 share more identity by descent segments with today's people from Western Scotland, Wales, and Northern Ireland because they or their direct ancestors migrated from these regions. But they didn't contribute substantially to later generations through mixing with local groups in Eastern Scotland. This scenario matches an emerging picture of West-East lifetime mobility of both males and females in the early medieval period in Scotland. Under such a model, it may be possible that there are indeed still undiscovered pockets of Eastern Pictish period ancestry. This would have been different from ancestry carried by BL03 and LUN04, and it contributed significantly to present-day populations from Eastern Scotland. Oxygen and strontium isotope analysis of teeth from these individuals holds promise to describe this further. Importantly, we also emphasize that randomness likely affected the pattern of identity by descent sharing in such a small sample size. Indeed, high variability in IBD sharing is observed among individuals from the early, medieval, and Iron Age groups. To some extent, this also appears between BIL003 and LUN004. Our results also show substantial identity by descent sharing between Iron Age, Viking Age, and present-day Orcadians. This supports our observations using allele frequency-based methods of strong genetic continuity in this region over time. Therefore, the marked genetic difference between Orkney and mainland Britain is not only a result of Scandinavian mixing, as previously thought, it's also pronounced genetic continuity that lasted for at least 2,000 years. The relatively low IBD sharing between BAL003 and LUN004 and modern-day Orcadians suggests the emergence of Pictish culture in Orkney wasn't linked with population replacement. Instead, it was largely due to cultural diffusion and connections. Identity by descent segments in Iron Age individuals from southeastern England are widespread throughout Western and Northern Britain. This is compared to the more recent Romano-British individuals from Northern England. The latter, however, don't share substantial IBD with any present-day people of the British Isles. The only exception is 6-Drift3, who was from the same genetic population as two early medieval individuals, I0159 and I1773, with Scandinavian and Northern European-like ancestry. 6-Drift3 also shares relatively more IBD segments with the present-day population from Scandinavia, Belgium, and the UK. This suggests that Scandinavian-like ancestry could have spread to the British Isles before the early medieval period. Social organization. 7 mitochondrial DNA genomes were retrieved at Lundin Links. This allows us to address questions about the Pictish social organization reflected in the individuals buried at the site. The use of the cemetery was relatively short, likely around 130 years. The individuals dug up were adults. The variety of mitochondrial DNA lineages was high. None of the individuals shared an immediate maternal ancestor. It's worth noting that individuals retrieved from the horned cairns complex show evidence of family links. This is based on the presence of highly heritable skeletal traits. These include congenitally missing teeth, torus mandibularis, and septal apertures on the humeri. However, the two individuals yielding mitochondrial DNA, Luanvowo1 and Lanor19, are not maternally related. In a matrilocal system, which is typical of matrilineal descent, low female postmarital migration and high male migration decrease female mitochondrial DNA diversity. This result suggests the individuals buried at Lundin Links were unlikely to have been practicing matrilocality. This assumes that this cemetery is a snapshot of the community near Lundin Links. Individuals did not return to their birthplace for burial after having lived in their marital community. Ongoing isotope analyses focused on the movement histories of the Lundin Links individuals are using strontium, oxygen, and other isotope approaches. These may further describe sex-specific mobility. Additional Y chromosome analyses will also help confirm whether patrilocality or neolocality was more common in Pictish society. 70% of matrilocal societies are linked with a matrilineal system. Insofar as burial treatment can tell us about the social organization in life, it's unlikely that the community at Lundin Links followed a matrilineal inheritance system. This interpretation challenges older arguments for matrilineal succession among Pictish rulers. However, while some individuals buried at Lundin Links may have been of elevated social status, the relationship between people buried in monuments like these and the Pictish uppermost elite is uncertain. The cemetery shows a wide variety of cultural practices. This is mirrored in the high mitochondrial diversity, suggesting relatively high levels of mobility within the Pictish social structure at this level of society. The burials are organized in complex and standalone graves. These are made of round and square cairns and long cysts. This complexity suggests that, as social practices influence the genetic structure of populations, the social status of archaeological sites can in turn bias our understanding of population structure. In this case, the samples may only represent a small proportion of the overall Pictish population. Non-harmonious kinship systems, like patrilocal and patrilineal or matrilocal and matrilineal societies, may also impact the genome in different ways. The lack of broad sample size and useful markers like Y chromosome to enhance kinship and mitochondrial DNA-based findings remains an obstacle. This stops us from illuminating further Pictish descent patterns. Conclusions. Overall, our data supports the current archaeological consensus. This argues for regional continuity between the late Iron Age and early medieval periods, but likely with complex patterns of migration, lifetime mobility, and mixing. We also show that Baal 03 and Lern 04 were genetically different from the pre-Viking Age Picts from Orkney. This suggests that Pictish culture spread to Orkney from Scotland mainly through cultural diffusion rather than direct population movement or intermarriage. We detect strong continuity between local Iron Age and present-day peoples in Orkney. but less pronounced connection between early medieval and modern people in eastern Scotland. More ancient genomes from the Iron Age and early medieval periods in the UK are necessary to clarify these relationships further, combined with analyses of lifetime mobility using complementary approaches like isotope analysis. On a more local level, our mitochondrial DNA analysis of individuals buried at Lundin Links is arguably inconsistent with matrilocality. This finding challenges the older theory that Pictish succession was based on a matrilineal system. This assumes that wider Pictish society was organized in such a way,